Good morning student. Today our topic is linkage and recombination. So first come to the linkage. As the name indicates, linkage means it is very close to one another. So you can define the definitions of linkage. You can define like this: the physical association of two genes in such a way that it inherit together to the next generation. That is the linkage. For example, this is the homologous pair of chromosome. Homologous chromosome. So in this homologous chromosome, suppose this gene A is present here, and this is the gene which is present, B gene is present here. So these two genes are distantly located, but they are the linked gene as they are present in the same chromosomes. Then here the crossing over, chances of crossing over will be more. That means after the gamete or during the meiosis, when the gamete will be formed, there will be more chances of crossing over here in this case. Then in the second case, suppose the gene is present here, A, and B gene is present here. So there is a less chance of crossing over here between these two genes. Less chance of crossing over between these two genes. Let's consider this third situation. Here, suppose this gene is present here, A, and the B is very close, and it means this is B. It is very close to one another. Okay, so basing on this location of the gene, your linkage is two types, that is complete linkage and incomplete linkage. In complete linkage, these two genes are very close to one another and there is no crossing over between these two genes. That's why they inherit together to the next generation, and recombinant frequency is very less. Then incomplete linkage. Incomplete linkage means if these two genes are slightly or it is distantly located from one another, then there is more chances of crossing over between these two genes, and the recombination frequency in case of the incomplete linkage is more. So we may say, or you can see the example. So complete linkage, you will find the grey body long wings in case of Drosophila, it is linked gene, or that is complete linkage is present here. Or incomplete linkage, yellow body, white-eyed, brown body, red-eyed, or yellow-eyed and miniature wings. These are the also linked gene, but they show the incomplete linkage. So we may say the linkage is inversely proportional to the crossing over. That means if the strength of a linkage is more, then the crossing over is less, and strength of a linkage is less, then crossing over frequency or crossing over is more. Clear? The next, the linkage group. What do you mean by linkage group? Linkage group means the gene present on the same chromosome. Suppose this is the gene present and other genes are also present. So all the genes present in the same chromosome, they consider as a linkage group. So we may say the linkage group equal to the haploid set of chromosome of an organism. Suppose we can write this linkage group equal to the haploid set of chromosome. Haploid set of chromosome of an organisms is your linkage group. For example, Drosophila, it is having the 8 chromosomes or 4 pairs. 4 pairs. In case of Drosophila, the chromosomes is 4 pairs or 8 numbers. So the linkage group will be the 4. In case of maize plant, that the chromosome is 20. That means it is 10 pairs. 10 pairs of chromosome. 10 pairs of chromosome present. So linkage group in case of maize, we may say it is the 10. So this is the linkage group. Clear? So next, your recombination process. Now here the T.H. Morgan T.H. Morgan study this linked gene in case of the Drosophila melanogaster, and while take the Drosophila melanogaster a fruit fly for its experimental sample, due to this following reason, this Drosophila melanogaster it grow in simple and synthetic medium. The life cycle of Drosophila H very short, that is nearly 2 weeks, and here the single mating produce a large number of progeny, and many hereditary variations which are present in case of the Drosophila. Again, the male and the female are clearly differentiate. That's why he took the Drosophila as his experimental sample and studied the linked gene, inheritance of a linked gene in this fruit fly Drosophila melanogaster. Again, he crossed this your yellow body. He crossed, Morgan crossed the yellow body white-eyed female with wild type of Drosophila and intercrossed the F1 progeny, and he obtained the offspring in F2 generations in the following ratio, that is parental type is 98.7% parental type of offspring are obtained in the F2 generation, and recombinant type 1.3% of offspring are recombinant in the F2 generations. Again, when I crossed the white body miniature wings with the wild type and intercrossed the F1 progeny, so in the F2 generation he obtained the parental offspring 62.8% and recombinant offspring 37.2%. So he proposed that if the two genes are very close to one another, then the parental combination is more. That means they are very close to one another, and they term the or they coined the term linkage for this parental combinations of offspring, and the non-parental or recombinant offspring produced due to the process of recombinations between the gene. So that is the recombination. So Morgan proposed the linkage and recombination. Linkage for this gene which are very close linked. That's why the parental combination is obtained, and recombinant and non-parental combination or offspring is obtained in the F2 generation due to the recombination, due to the recombinations of the gene. Clear? So this is the linkage and recombinations. Now comes to the gene mapping process. Gene mapping. Here Alfred Stewart went. He mapped the gene on a chromosome. So what is mapped the gene on a chromosome? That means this is the chromosome, and the location of a gene on a chromosome is called as the gene mapping. Location of a chromosome. Clear? So location of a gene on a chromosome. Location of a gene on a chromosome is called as the gene mapping, and the unit of your gene mapping is map unit and centimorgan, that is called the centimorgan. And what is the basis of your gene mapping? That is the frequency of crossing over is the basis of gene mapping. For example, if the gene are distantly located, that is this A and B gene are distantly located, that means the crossing over frequency will be more. So if the crossing over frequency is more, then we can say that the two genes are distantly located. If these genes are or crossing over frequency is less, then we can say these two genes are very close to each other in the chromosome. Clear? So that is the basis of your mapping for our your basis of gene mapping. What is the basis of gene mapping? Recombination frequency. Suppose for example your questions come like this. If the gene, suppose your A and B, it is having the 20 map unit, and A and C it is having the 10 map unit. So find out the your location of a gene on a chromosomes. So suppose this is your chromosome, and as the AB is 20 map units, suppose this is the A and let's say this is B. Suppose this is A gene, this is B gene, and here it is 20 map unit. 20 map unit. So then AC equal to 10 map unit. So AC 10 map unit. Here we have not given that the C is present in which direction. So the C can be present like this here. That is C, or it may be the C also. So there is the two possibilities that the gene may be like this: A, C, and B is one possibility. Or another possibility, suppose this is 10 maybe it is the 10. So C, A, and B. C, A, and B. This is the another possibility. So that means you can trace the location of the gene on the chromosome. So that is called the gene mapping. Clear? So today we discuss about the linkage, crossing over, and gene mapping. Okay, student? So clear all about the linkage, crossing over, and gene mapping. It is very important for your examination point of view. So go through it, read properly. Go through the NCERT book also. Okay, students? Thank you.